Afternoon, everyone. Indeed, a pleasure to be here with you all. I think you can reserve some after the session, I hope. Thank you so much. So I'm going to talk quickly on standardization of herbal medicines, which after my medicine, I did a PhD in standardization. I got a patent on that. So I'll be quickly going through the key regulatory requirements for it, which will be useful not only for individual practitioners as well as for people who are hoping to manufacture their products. Maybe this again is not in the text. You can contact me in case if you wish to get some more details about it. The content is going to be in general introduction, the summary about it, the challenges which are faced, and I think some of the previous speakers mentioned about some of the challenges, about why there's a requirement of standardization, why there's a requirement of safety amongst the herbal medicines. Not that we doubt about the safety and we know about the efficacy of the molecules, but it's required when you want to compare it with the modern medicine drugs. Limitations which we have and procedures in which the standardization needs to be done as per the guidelines. I'll be specifying morely on the WHO guidelines here. That's what is the last part of the session. So when we talk about the transition in herbal medicine, we have seen that earlier the herbal medicine used to be a individualized herbal medicine, maybe in different parts of the world, which has now become a generalized herbal medicine, which has been manufactured in a huge quantity in the in different manufacturing firms and is given to the people who consume it. So the challenges when you see here are specifically the availability of the raw material which has been used. There were some questions which were earlier raised about whether I need to use the seaweed. I think the session was on the seaweed, whether I need to use the seaweed as a raw material or whether I need to do an extract and use it. What is going to be the difference? So that's exactly the point which comes here. The authentication of that raw material, how is it grown? Where is it grown? Where does it come from? How did the person who is creating it has received it? Proper standardization methodology on how you are going to standardize it and use it as a fingerprint when as a manufacturer or as a regulator, you're going to check whether it's the same medicine which is being used in the final product. So it can be a single drug, a single drug, crude drug, or it can be a formulation which can have multiple herbal drugs in it. And of course, the quality control methods, which will be the stability studies, etc., which needs to be in place to see how much duration the drug is going to be stable for how much time when you're going to give it to the patient. So key few things, I'm not going to read across, the slide deck can be made available if required. What is essential for standardization is the quality of that last formulated molecule which is out to the consumer. The active principles or the constituent which is going to be given, either it's in the extract form where you extract it, or it's going to be the crude form which you're going to give it to the patient. The physical, chemical, phytochemical standardization, quickly I'll share with you what are the different ways how it is done, and the in vitro, in vivo parameters which are used. So either you're going to do a toxicity study if it's required, if you're going to extract it, or you're going to use it as it is if it's going to be a plain herbal, which you need to give. So the task is about the quality. Talking about the challenges, not going to emphasize more. We have heard a lot of the challenges about it in the earlier sessions. No official standard, though we have pharmacopoeia, there are a lot of formulations which still don't have standards. And there are challenges and queries are being raised saying whether it's a quack medicine or whether the medicine which you're giving is really useful or not. Manufacturers are doing it. They are having some kind of testings of the formulations which are there, which is their own parameter. Whether that parameter is available in the pharmacopoeias with the regulatory authorities, we have queries there. Difficult to identify the presence of all the ingredients. If it's a formulation, there'll be more than one ingredient which is a part of it. So how to identify which ingredient is there in that formulation is again a challenge in herbals. Different ways, you can see the fingerprinting on the left-hand side, HPTLC fingerprinting, which has been conducted here, or the microscopic analysis which have been done, which is useful for identifying the physico-chemical properties of that molecule, which we are going to estimate. Okay. Advantages are N, number of advantages which we have. You ask any person, around 10 people out there, I'm 100% sure that at least 4 people will tell you they have used herbal medicine in their life. So it cannot happen that there is no one who has not used herbal medicine. There's a long history. used everywhere. Of course, availability of the raw material is there. You don't need any big manufacturing units to make it available. You should need to know, authenticate the proper raw material, and then you can use it. Now, herbal medicine, potent medicines specifically used all across with different names, different cultures. Limitations, quickly, ineffective in acute medical care. And that's the main important drawback which the herbal medicines all across have. And that's why wherever you hear about it, you hear that, yes, it's used in chronic diseases only. But of course, I would like to say that there are certain drugs which are used even in acute conditions, though they are not being, you can say, published very much that it can be used, but there are certain. But presently, this is the main limitation. Inadequate standardization, already mentioned, and lack of key scientific data. Whether we have any clinical studies, randomized, double-blind clinical studies about it with the N number estimate, no. If we are supposed to do it, where's the finance which we are going to get it? Is any of the university or institute going to finance this kind of work? Is one of the challenges which has been faced by majority of the people who are treating the patients. So there's a need which we need to do so that the patient or the consumer is aware about the safety. Need to have a standardization. Only on this point I want to emphasize this batch to batch variation exists still in the end products, and that's one of the challenges which are being raised and pointed out by the modern medicine. When we say standardization, it is the entire field of study, right from the plant, collection of the plant or the crude material, to its clinical application, which goes to the customer. Definition as per WHO, it refers to the drug's identity, what botanical name, what it is, determination, purity of that material, name of the adulterant in case if you have, so we need to ensure there's no adulterant in the end product, and all the other various parameters, biological, physical, chemical, microscopical, etc. Different current regulations and the pharmacopoeias available, you can read it on the slides. These are some of the ones which are anyway available within the scope. When you mention about a standardization of a drug, it consists of not only as a drug, it has its own biological activity, its chemical profile. So when you isolate it, you are able to understand whether it's a flavanoid, whether it's, what kind of active material is there. Now, in a herbal drug, it becomes very difficult to identify whether the identified material or the identified constituent is the active constituent or not. So that's one of the other challenge which has been faced by the herbal practitioners or the people who want to do a quality aspect on this. So in short, when you see a methods of standardizations, there are 4 key pillars. Of course, you have botanical, macroscopic and microscopic analysis. You have physical, where you need to conduct all these tests. Again, it's there published. All the publications are there on the slide. So you can refer to those full text articles. Biological and chemical. These tests are compulsory and needs to be conducted so that a drug becomes a drug and not a food supplement. The processes, again, as per WHO, are quite vast, not going in details. I have just 15 minutes, 5 minutes I'll keep for some questions in case if you have. These are the detailed tests which needs to be done under every window to make it a standardized drug. Okay. Again, the WHO guidelines mentions these key parameters. Sorry, it's quite small. The first is about the reference of the identity, microbiological test, toxicity studies, reference to the pharmacological parameters, and reference to the physiochemical parameters. That's the fingerprinting, which I'll be talking about. Now, this is one of the best method now, which you can say is available to us, which is the chromatography. I suppose everyone knows what's a chromatography. It's a plate which is run across and is seen in the UV to see what all the ingredients are there a part of. Some details are mentioned in the slides for people who like to go through it. Now, this TLC or thin layer chromatography, as it's called, is an easy way initial screening and evaluation of this. Now, you can see few photos and then we end the presentation. So you have this quantitative analysis on TLC, which has been run across for a plant. This is how it will be seen. The other instrumentations are HPTLC, which is the first one, which you have seen as a fingerprinting. That is high performance thin layer chromatography. It's an instrument where you can run and try to isolate the compounds. The other is LC, or the liquid chromatography and mass spectrography. It's again another instrument where you're able to view and analyze it, becomes a good choice if you want to do a drug development. So that's another one. You have LC-NR, NMR, as well as gas chromatography. Gas chromatography is used when you want to visualize and find out the volatile constant. So all the oils extraction is done by GC, and GC-MS is the way how you measure it. You have GC-FID and you have SFC. Again, the another of instruments where you can analyze and extract it. DNA fingerprinting is also nowadays done, so it can become a good way of understanding the actual DNA content of that plant, and genetic markers. So this a little bit of 5 slides gives you all the instrumentation which is available right now for standardization, which can be used for a manufacturing unit or for people who really want to put it as a product into the regulatory affairs windows. So you can see here one molecule, which was my research molecule, which has been run across to see the extracts which have been done. One is the alcohol extract, another is the ether extract. You would have heard these are the extracts which are isolated and given in the capsule form to the firms to the patients. You can see the variations also with this HPTLC and HPLC extracts which have been done here. You can see the difference here very clearly in the plants which have been collected in the month of May and in the month of December. And there is a difference in the fingerprinting and the plants. The collection time of the plant, which has been mentioned in Ayurveda, is very important when you consider this. And this is the last slide of me. I'd like to thank 3 people, my guide and mentors. Got an opportunity to work with Professor Hilbert Wagner. I suppose people here might be aware of the name. Is quite well-known person in phyto, so I got honored I could work in his lab in Munich. Dr. Sane, through which who I did my post-graduation from medicine, changed to science to do a research in this. And nephrologist in one of the reputed hospitals in KM, where I did my clinical study after isolation in the capsular form, a nephrologist with which I could show a change in the patient. So 3 people whom I would like to thank in this session. That's it. These are again the references which have been used across in all the way. There are much more references. You can connect with me in case if you're interested. That's it from my side, I think.